Okay, so let me tell you guys a couple of things as we are getting started. First, we won't quite finish this set of notes today. You're going to be tested on it, but I will test most deeply on the stuff that we got to discuss. What's going to happen is I will post a little 20-minute catch-up lecture at the end, and then I'm also going to post a homework review quiz for you guys that will have questions over the catch-up lecture, right? So I'll give you a decent indication of what kinds of things. And the catch-up stuff is stuff that you've already done in lab, so I'm going to reinforce the stuff that you've done in lab. I think my plan is that I will get the catch-up lecture up today or tomorrow, and the homework quiz you'll have until, I'm thinking Sunday to do it, so that all of the answers will pop up and you can discuss it on Monday with people if you need to. Remember that the quizzes are collaborative and they're open books, so talk to as many people as you want and work on them together. Good? Okay, what else do I need to tell you? After today is fall break, and I won't see you again until Tuesday after fall break. Isn't that right? What? No Thursday class, yes. Did you just get happy? Okay, so no class for a week, basically, right? Does that make sense? And then what happens when you come back on Tuesday? Yeah, you have an exam, and the exam only covers 2 body systems: endocrine, which is massive, right? And then muscle, right? So you guys should be okay on both of them. Everybody good? Go ahead.
Sorry, one more time. When is that quiz going to open up?
Tonight or tomorrow. I've almost got it written. I just need to make sure I've got a couple of questions over the catch-up material. But tonight or tomorrow. Good? And I know that there are some level of complaints when I give you guys homework quizzes over weekends or breaks. But 2 things about that. First, I am writing them for you so that you can assess whether you know the material, whether you expected questions of this type. I'm not writing them to torture you. I'm writing them to help you. And also, you are a college student. So the expectation of no homework over fall break is kind of inappropriate for this situation. Okay, so what we did last time was this. We basically looked at the mechanism of sarcomere contraction, right? Sorry, my coat is not free. So we looked at the mechanism of sarcomere contraction, but what we didn't do is we never really put together action potentials and how they cause sarcomere contraction. So that's what we're going to do today. So the plan today is that we are going to finish sarcomere contraction and relaxation, and then we're going to do that worksheet. Did you bring the worksheet? But we're going to do the worksheet in pairs of 2 or 3 so that you can consult with the other smart people that you know. So right after we finish this and talk about rigor mortis and all that kind of stuff, I'm going to ask you guys to get in little groups. Because if you have to do it individually and I call on you, it's pretty stressful. But if you have to do it with a friend, collectively, you are brilliant, just brilliant. Okay, so where we are going to go is we're going to go back and tell you a little bit more about our friend actin, right? So we went through the cross-bridge cycling before, but we really never addressed these 2 proteins that you can see right here, this long skinny protein or this other one. So we need to go back and put that together. So let's remind ourselves what actin looks like. Where are you, actin? And then we'll add in some details to the structure of actin. If I can find actin. Okay, so we did not address last time troponin and tropomyosin. But you probably talked about it in lab. Did you talk about it in lab? Okay. If you didn't, that's fine. So we know basically how sarcomere contraction and cross-bridge cycling goes, but we don't know how to initiate it. So I have to tell you something about actin's default state. Actin's default state looks like the top, in which the active sites, the actin active sites, are actually blocked from cross-bridge formation. Do you see that? So even if a myosin head wanted to attach to that little black dot, which is the actin active site, it couldn't do that. So what we are going to do today is figure out what these proteins have to do with initiating contraction. So here's tropomyosin, the long skinny one. I think of that protein as the barricade protein, okay? It's barricading cross-bridge formation. As long as it's there, you're not going to get any cross-bridge formation. Is that okay with you? Okay. And then this little guy right here is called troponin. And troponin is the switch for the barricade. Okay? So what we are going to see today is how you get the switch to be activated and move the barricade so that you can start the cross-bridge formation and the cross-bridge cycling that we already learned. Got it? Okay, so what we're going to have to do with that is we're going to have to go back and say, okay, we already know that skeletal muscle fibers, which we're talking about here, will not contract without nervous system stimulation. Yeah, we already know that. But we don't really know how nervous system stimulation goes. So let's learn a little bit about that. So specifically, you guys remember that a neuromuscular junction is the site. Yes, here's a neuromuscular junction. The site at which a neuron communicates with a skeletal muscle fiber. Okay, so a neuromuscular junction is really just a synapse. And you guys learned about synapses in anatomy. We're going to learn about them in detail right after this. This is a certain neuromuscular junction is just a synapse between a neuron and a muscle cell, right? Got it? Okay, so this is the neuromuscular junction. What's going to happen is an action potential is going to come to the end of the synaptic terminals of this neuron. And by the way, guys, this neuron, since it stimulates a skeletal muscle, can you tell me what kind of neuron it is? Is it somatic or autonomic? It's somatic, right? And is it motor or sensory? Right, so this is a somatic motor neuron, yes? So a somatic motor neuron forms a junction at a skeletal muscle, and this junction is actually going to be called a synapse. And the specific type of synapse is a neuromuscular junction. Okay, remember skeletal muscle fibers really don't do anything unless the nervous system tells them what to do. So what is going to happen is we have to start with the nervous system before we get a skeletal muscle response. Got it? Okay, so we're going to start with the nervous system. And so what's going to happen is the nervous system portion starts the excitation. We get an action potential. We know how action potentials work, right? Started at the cell body, axon hillock, action potential. Then what is going to happen is neurotransmitter release. Neurotransmitter at skeletal muscle fibers is always the same one, so it's easy. It's always acetylcholine, okay? The mechanism of neurotransmitter release we haven't learned in detail yet, but I will tell you that action potential gets to the end of the axon. At the synaptic terminals, what's going to happen is you have these new channels that you haven't met before. These new channels are called voltage-gated calcium channels. And what's going to happen is they open in response to depolarization. Calcium moves in, triggers exocytosis. Does that make sense? Now, even if you haven't heard of the voltage-gated calcium channels before, action potential, exocytosis, and acetylcholine. Got it? So action potential, exocytosis of acetylcholine. All right, so once we have acetylcholine released, what we are going to have is the acetylcholine is going to bind to receptors. And the receptors, you can see them right here. The receptors are on a portion of the muscle cell membrane that is really, really curved, called the motor end plate. Anybody want to hypothesize why the motor end plate would be highly curved?
More surface area.
More surface area, more room for receptors, faster response. Yes? Does that make sense? So, dump acetylcholine, bind to the receptors. The receptors are also ligand-gated sodium channels. So when you bind to them, yes, you're going to open these ligand-gated sodium channels, and sodium is going to move which direction, guys? Into the cell, causing blank polarization. Depolarization, exactly. Right? So what we are trying to do is we are trying to get this muscle cell to have its own action potential. Yes? So the muscle cell can have its own action potential when you hit threshold. Threshold is not exactly the same number in muscle cells as it is in neurons, but it's definitely the same part of the story, right? So once you hit threshold, you're going to get an action potential. But remember that the action potential is not going to be limited to the cell membrane in muscle cells because you have these tunnels that tunnel into the cell, right? What are they called?
T-tubules.
T-tubules, exactly. T-tubules or transverse tubules. And the T-tubule, by the way, guys, is really a continuation of the cell membrane, and it has voltage-gated ion channels on it, so the action potential can be taken into the middle, right? So that's a T-tubule or a transverse tubule. So let's put this together with how this will cause contraction. Do you guys remember that I told you that the T-tubule runs right by the sarcoplasmic reticulum. It's right next to the sarcoplasmic reticulum. And the sarcoplasmic reticulum stores what? What is the SR? Calcium, yes. Okay, so here's the story that we're going to tell. The action potential gets started if you hit threshold. It goes along the cell membrane, but isn't limited to there. It goes down the T-tubule, yeah? And it causes release of calcium from the SR. The way that it causes release of calcium is information that I wasn't taught in physiology, and it's not particularly important, but I'll tell it to you anyway. There are these little receptors that actually link the cell membrane of the T-tubule with a calcium channel that's on the SR and cause calcium to get dumped. But I want you to think about where calcium is going to go once it gets released. And calcium is released from where it's sequestered in the SR. Which fluid is it dumped into? ICF or ECF? It's dumped into the ICF. And do you remember that the SR wraps around all of the myofibrils, so it's like snuggled up to the outside of the myofibrils, right? So what I'm doing ultimately is dumping calcium very near my sarcomeres. Yes? Okay, got it? Okay, so I'm going to dump calcium really near my sarcomeres. I'm about to show you guys an animation in just a second, but let's go back to this picture. Okay, so I'm going to dump calcium near my sarcomeres, and the calcium is going to go and bind to the switch for the barricades. Yes? That's what we were trying to get to. So the calcium binds to, which one was the switch again? Do you remember? Troponin. And when you flip the switch, you move the barricade protein called what? Tropomyosin out of the way. Yeah? Is that with me? Are you with me? Okay, so when you move the barricade out of the way, do you have to encourage myosin to bind to actin active sites? No, it wants to, right? So what it will do is, remember our thing that we did, right? You will form a cross bridge, yes? Everybody hands up. Cross bridge formation. Does this cost me an ATP? No. How do I initiate the power stroke? Phosphate falls off. Power stroke. What happens here? ADP falls off. I'm stuck here unless I have a what? ATP. ATP binds. Yes? Have I re-energized my catapult? What do I have to do? Hydrolyze ATP to ADP plus phosphate. Got it? Okay. Right. So that can only start when the tropomyosin gets out of the way. Does that make sense? And I'm going to continue doing that as long as I have calcium out of the way, or calcium present. Because with calcium present, you're just going to be able to reach the next one and the next one and the next one and the next one, right? So let's look at that. So you got action potential moving down the T-tubules, yes? You got reaching the sarcoplasmic reticulum. You got release of calcium from the SR, right? You got exposure of the active sites because calcium bound to which one again? Troponin, and moved what out of the way? Tropomyosin, exactly. And now you start your cross-bridge cycling. You are going to continue to do cross-bridge cycling until calcium goes back where it came from, right? So let's look at an animation, and I'll show you how you get rid of calcium. Ready? Okay, so let's look at a couple of animations, starting this whole story. Ready? So with the first one, okay, this is my somatic motor neuron, and it is going to have an action potential. That's the voltage-gated calcium channel that I told you guys about. When calcium moves in, it triggers the release of exocytosis. It's going to diffuse across, what is the synapse called again? Neuromuscular junction. Good. It's going to diffuse across the neuromuscular junction, and it's going to bind to the acetylcholine receptors that are also what kind of channels? Ligand-gated, not calcium, sodium channels. Good. Calcium would work too. It just doesn't exist here. But calcium would also be depolarizing. And then as calcium moves in, right, what I'm going to get is depolarization. When I hit threshold, yes, which is just represented by a little zap here, then I'm going to open all of the voltage-gated sodium channels along the cell membrane. Got it? Okay. Then that's going to lead to this. Ready? Come on. Okay. So action potential, it's going to go down. What are those probably? T-tubules. What is this? Right next to the T-tubules, that's probably what? SR, sarcoplasmic reticulum. So the action potentials go down the T-tubules. It will ultimately cause the release of calcium from the SR. Watch what calcium does. Diffuses into the nearby sarcomeres. And then what's going to happen? Blow it up. Watch where calcium goes. So I've got my tropomyosin barricade. I've got my troponin switch. And I've got my actin with the covered up active sites, right? Calcium binds to the switch. Got it? Barricade moves out of the way. Cross-bridge formation is going to occur. Get up there. Do it. Do it. Do it. Cross-bridge formation. They didn't show the phosphate falling off, but the phosphate will fall off. Yes? And it's going to be stuck there unless, of course, I have ATP to bind. Does that make sense? So go ahead.
So is the same calcium on this one the same?
No, that's a fantastic question. So we haven't talked very much about this calcium, but we will in the next set of notes. So this calcium is extracellular calcium, and it goes extracellular to intracellular on the presynaptic cell. This calcium goes sarcoplasmic reticulum to intracellular fluid, and then you'll see in just a minute, we're just going to put it back in the sarcoplasmic reticulum. So one of them is extracellular calcium, and the other one is exclusively intracellular calcium. So that's a good question. Go ahead.
Can you remind me, for the action potential that goes down the axon hillock, is that going to be also stimulated by acetylcholine in the beginning?
So you mean the one that occurred previous to this action potential? Well, it could be. It was stimulated by any kind of stimulus, but let's hold it to a chemical. The chemical could have been acetylcholine. It could have been another neurotransmitter.
Okay.
Yes? So, but ultimately there was a ligand down at that end of this cell that started the action potential on that cell. Yes? On muscle, it's easy because it's always acetylcholine.
Okay.
On neurons are specific to specific neurotransmitters, pretty much. Does that make sense? Okay. All right, so those are good questions. All right, so now, here's my question to you. How do you ever get this thing to relax? So what I'm going to have to do in order to make it relax is I'm going to have to ultimately get rid of the calcium. But then don't I have to stop doing this, right? Stop doing that, stop releasing calcium, and then get rid of the calcium that was already there? So let's just look at how you would dependably cause this muscle fiber to relax. So first thing is that you shouldn't have any more action potentials on the axon, or else you're going to continue to release acetylcholine and continue to depolarize the postsynaptic cell. Yes? That makes sense? So you got to stop stimulating it here. Does everybody agree? If you don't stop stimulating it here, you won't get it to relax. So that's what I'm saying. So you have to stop the action potential on the axon. And then the next thing is, what about the acetylcholine that was already there from the last release? As long as that's around, don't you continue to depolarize the postsynaptic cell? If the acetylcholine stays around, it continues to open the ligand-gated sodium channels, and you continue to depolarize. You guys with me? So you have to get rid of the acetylcholine. That's the important thing. So there is a mechanism of getting rid of acetylcholine that I will show you guys. So the way to get rid of the acetylcholine is an enzyme that is present on the postsynaptic cell membrane, and it's an easy one, and it shows you right here. I just didn't show you yet. Watch. It's going to cause the response desired, right? And then note that what's going to happen is the acetylcholine is going to pop off, and it's going to interact with an enzyme that is present on the postsynaptic cell membrane. That enzyme is called acetylcholine esterase. I'll tell you how to remember that in just a second. And here's what happens, guys. The acetylcholine esterase will break down acetylcholine into acetic acid and choline. Choline is a B vitamin derivative. It will reuptake the most precious of those two, which is choline, to make more acetylcholine out of it, right? But it's not going to activate the receptor any longer once you change the shape, right? Does that make sense? So present on the postsynaptic cell membrane is acetylcholine esterase. The way that I remember what acetylcholine esterase does, it's acetylcholine erase. Erase. But then you can spell it, right? Because it's just the word acetylcholine, right? Erase. So acetylcholine esterase, okay? That's not what the name means at all, but it's easy to remember that way. Okay, so that's acetylcholine esterase. So once acetylcholine esterase gets rid of the present acetylcholine, then what you have to do is you won't have action potentials if you don't have a graded potential, yes? No graded potential on the muscle cell membrane means no action potential on the muscle cell membrane. But now the last thing that we have to deal with is we have to deal with the calcium that was already released. Because can you agree that as long as calcium is present, you are going to continue to allow for cross-bridge formation? Yes? So how do you get calcium to go back where it came from? Any idea? You have to pump it. Yes. Why do you have to pump it? Is it because of size or charge, or is it because of what? It's concentration gradient, because there's calcium stored in the SR. And I released a little, but I have to put it right back into the storage facility where there's a boatload of calcium, because it didn't release all of it. So I have to pump it back, which is interesting because it's probably the first time that you've ever seen an intracellular pump. That's not the last time you're going to see them, but they usually, when you meet pumps, they're on cell membranes, right? But this one is pumping from what fluid to what fluid? From, not interstitial, huh? ICF to, I don't know what you call SR fluid. Let's call it SR fluid, okay? ICF to SR fluid. But note Mike's question from before, it doesn't ever go extracellular, right? I mean, it could leak out, but it's in this story, it's not ever going extracellular. Is that okay? Yeah. So once you pump the calcium back into the SR, then your switch is no longer flipped. And what happens to tropomyosin? Slides back into position. When tropomyosin slides back into position, the actin, the myosin head can't contact the actin active site, and the actin slips back into the relaxed sarcomere position. Does that make sense? And it stays there unless you start the whole process over again. Everybody good? Go ahead.
So what causes like a cramp?
We've got cramps in just a minute. You want to hold on to them? Okay, go ahead.
So if you don't have, since it takes energy to also stop this process,
Yes.
Is your body gonna like stop doing an action, like a muscular action, before it runs out of energy? So
Not usually, because we will see that you have a whole bunch of storage mechanisms, a whole bunch of plan B, C, and D with a normal healthy muscle. If you have an ion imbalance, if you die, we'll talk, rigor mortis is the next thing we're going to talk about. So why does rigor mortis do that, right? It's directly related to the story that we're telling. So let me answer that by showing you this. Okay, you guys know what rigor mortis is? Yeah? It's the sustained muscle contraction that happens after death. Usually, it just holds you in the position that you died in. So if you died flat on your back, you're really going to go into rigor flat on your back. But if you died like a lot of people do, you should eat your fiber and take care of your heart on the toilet, right? Then you actually go into rigor in this position, right? Which makes it pretty hard for people to get you out of teeny tiny bathrooms. So the problem with being dead is that there's no ATP, I mean, among other problems with being dead, but it might be pleasant. I've never done it. No ATP is being made after death. Why? Why is no ATP being made after death? Because oxygen, right? Oxygen is really the rate limiting factor for this whole ATP production. And so what happens, remember, where do we get stuck without ATP? We did this, that was free. We did this, that was free. We get stuck right there, right? And then add to that the fact that after you die, it's going to be hard to keep calcium sequestered in the extracellular fluid and in the SR, so it's going to start to slowly leak in. Does that make sense? Down its concentration gradient. So I'm going to form more cross-bridge formation over time, right? And then it all gets stuck there. It doesn't get stuck so much that people are dancing, but it gets stuck in the position, right? So what happens is the calcium gets stuck. And calcium, not only are you stuck here, but you can't get rid of the calcium without ATP, can you? Because how do you get rid of calcium? You pump it back into the SR. So I'm getting stuck, I'm getting slightly more stuck, and then calcium is leaking, and I can't get rid of the calcium. So what I get over time is a progressive sustained muscle contraction, right? Does that make sense? Until I get into the position of rigor, and this is rigor. So here's rigor, okay? So that would take a decent amount of muscle contraction if you were alive. Can we agree? To keep your legs up like that for long periods of time. So what you have is rigor. This person probably died on this actually came from a physiology journal. I know it looks just like it came from rotten.com, but it actually came from a physiology journal. So that's rigor, and rigor tends to hold you in whatever position you died in, but it's going to leave as protein denaturation starts to happen, as your proteins start to break down. So how long does rigor last? Well, it's really temperature dependent, right? But usually 12 to 15 hours. If it's really hot, it's going to go through the whole process way faster, right? And if it's really cold, it's going to go through the whole process way more slowly, right? Now, rigor ends up being significant to medical professionals and in some cultures, because in some cultures, and I'm ignorant of cultures that are too far different than mine, but I do have people who will educate me about such things. First of all, in some cultures, including Orthodox Judaism, you have to be buried within 24 hours. Can you see how you would run into people in rigor if you had to be buried in 24 hours? Yes? Okay. And apparently in Somali Muslims have to be buried within 24 hours, culturally, religiously. So you can run into people in rigor, and this means that the people who are preparing people for burial can have to deal with rigor in order to get you into a casket or whatever you use. In addition, with medical professionals or EMTs or funeral directors, if you have a person who died in a position, your muscles do not let go. And if you can't get the muscles to let go and you have to get someone out of the bathroom, you might have to break bones, right? Instead of doing that. So they just don't let go. But you can't just let them sit there until they denature, right? Typically not plan A. So that's rigor. And rigor has directly to do with the story that we were just telling. The other one that I want to cover is polio. Okay, so poliomyelitis has been almost completely eliminated from the world, but we do have little pockets of polio popping back up. In all likelihood, you guys were not vaccinated against polio. My mom was vaccinated against polio, but I wasn't, because polio has been so rare for so long because there was an incredibly effective vaccination program for polio worldwide. Here's what happens with polio. It's a virus, and it destroys motor neurons. Somatic motor neurons is what we're talking about here. And if you destroy a neuron, is that damage likely to be temporary or permanent? Permanent, because most neurons are amitotic, right? So what you get is permanent. What's the impact on the muscle if you destroy the motor neuron? Paralysis, right? The muscle can't contract in the absence of a command to contract. So you have permanent paralysis. You can have complete paralysis depending on how many motor neurons you lost. You can have partial paralysis and muscle weakness, right? And so what polio would do is cause partial to complete muscle paralysis. If it happens in the diaphragm, you're going to be in an iron lung. Just how one of the treatments historically for that. We, of course, historically, polio is really significant for a lot of reasons, but here in the United States, we had a president who had polio. Who's that? FDR. It was FDR. He had polio and he had partial paralysis, but he still had the ability to walk and stand. And he was always photographed with other heads of state standing, because, height, power, perspective. So now polio is actually having a little comeback. It hasn't really impacted this country that I know of very much, but it's getting little blooms in parts of the world. This little boy had polio, and you can see the muscle atrophy that eventually results, and then the bone atrophy that eventually results. And so polio is pretty completely preventable by vaccines, but guess what's happening? People are, well, it's not here in this country because we're not really vaccinated against polio, but worldwide, people have this perspective on vaccines that is reducing vaccination rates and bringing a big comeback of preventable diseases. So this dude has a comment on that. This is polio, vaccinate. And then look at that. Don't you want to hang out with him? Because that's funny. 0.0. I'm like, you got a good sense of humor, dude. So, yeah. All right, so as far as cramps, what cramps are, and then I'll do just one more. I'm not going to do all of these disorders because we're in a little bit of a time crunch. Cramps are when action potentials on motor neurons fire at abnormally high rates. That's often due to an ion imbalance, which will cause things to be slightly depolarized. So it could be a calcium imbalance or a potassium imbalance, and it causes involuntary tetanic contractions. The other thing that can cause cramps is damage to a muscle, like a Charlie horse, which will cause the ions to leak out and have a localized ion imbalance. So that's what it has to do with. And it's usually just due to an ion imbalance. The ion imbalance could be due to a hormonal imbalance, but a lot of times it's just due to what you're eating or not eating or drinking or not drinking. Okay, 2 more disorders and then we'll go to do the worksheet. Ready? Okay, this one is an interesting one. Actually, I'll do this one first. Myasthenia gravis is an autoimmune disease, and I want to tell you what happens with myasthenia gravis. With myasthenia gravis, what happens is your immune system causes blockage or destruction of the motor end plates. So motor end plates are where the receptors are, yes? Okay. You get exposed to some pathogen, I don't know which one, and what happens is you make antibodies which either destroy or block the motor end plates, and that means that this muscle cell, if you can't stimulate it, is going to be paralyzed, yes? But the paralysis doesn't have to be permanent because you're not killing off the cell. If your immune system ever lets up, then the cell can remake or reuse the receptors. Does that make sense? So with this, how myasthenia gravis tends to manifest is this sort of roving loss of muscle contraction, like Bell's palsy in the face, right? Or muscle weakness in one side, or sometimes a little trouble breathing, or a little limp in the gait. So that's myasthenia gravis. Like all autoimmune diseases, there isn't a cure for myasthenia gravis, but it doesn't necessarily have to have a completely negative progression because you can go into remission if your immune system just backs off for a little while. So that's myasthenia gravis. And then the last one is this genetic disorder that you've probably heard of called muscular dystrophy. Have you guys heard of muscular dystrophy? Muscular dystrophy, there's several different forms. Generally genetic. The one that you're probably most familiar with is X-linked recessive. And with an X-linked recessive disorder, which sex will manifest it most frequently? Males, right. Because you've only got one X chromosome. Everything on your X chromosome is potentially expressed, okay? So what happens with muscular dystrophy is it causes a weakness in a cytoskeletal protein connection. And so remember that you pull on the cytoskeleton to make muscles contract, and you're going to continue doing this over time, and it causes a progressive deterioration of the muscle fiber. Do you think that exercise helps that situation? No, it doesn't. And it also does it in cardiac muscle. And you can't stop using your heart, and you can't stop using your diaphragm. So this is usually diagnosed, this is the form called Duchenne muscular dystrophy, and it's usually diagnosed in the toddler ages, usually because a pediatrician that's paying attention notes that your baby has a pull-up pattern that's different than other babies because of muscle weakness. So usually when babies start to pull up, they just climb up things and they use their legs, right? But with these babies, they've got a lot of muscle weakness. And so what they do is they tend to depend on structures more. So they pull themselves up kind of Frankenstein. And then it ends up causing differences in gait as they age because of this muscle weakness that you get. And sometimes these little boys live into adulthood, and sometimes they don't. It really depends on whether it dramatically affects their heart or their diaphragm. But you're going to have a progressive deterioration of the muscles. Okay, we'll do tetanus and botulism and the other ones when we do synapses. So we'll come back to those. So what I want to do now is I want you to get out that worksheet, and I want you to get in groups of 2 or 3 people that you feel comfortable talking to. And then we are going to run through the worksheet as a group. Okay? Please lower your stress level. I am not testing your knowledge. I'm just trying to get you to think about and use these words. You're going to do it. Feel confident. Everybody with their people? Okay, so here's the plan, okay? First off, deep breath. I apologize in advance to all of you introverts who hate to talk in class. but part of the way that you get good at this is getting used to using the language, right? So that's what we're going to work on here. This worksheet has really all of the complicated parts of muscle contraction and relaxation in a way to practice, okay? So here's what I'm going to do. I am going to start at the back, okay? And I'm just going to call on little groups. Try not, if you were that group, try not to call out the answer. Give them just a second to sit with it, because sometimes the word doesn't come up instantaneously, but we can be patient and wait for them for just a second. This is not actually testing your knowledge, okay, because you just learned some of this stuff. If you need to look it up or talk to each other, do that. Cool? Okay, so, and I'm going to give you a lot of flapping around signals for what you should be doing. All right, you ready? The functional unit, that we are at this one right here. I'm going to try to keep you guys on track so the other guys got lost. The functional unit of skeletal muscle fiber contraction is a, look up here. It is the thing that contracts, and it's from Z line to Z line, and it has the actin and the myosin in it. It's that repeating pattern. What is it? No. What is it? Sarcomere. Yes, it is a sarcomere. Good. Okay, now, next group over here on the corner. Okay, Karina, and I can't see your face, but hi. So the theory, the only thing that we have learned the entire semester that is attached to the word theory, the theory that explains skeletal muscle contraction is called the blank blank theory. What are they? It's, what are these things doing past one another? Talk to each other. You two, you're a pair. You're a pair. The blank blank theory of muscle contraction. Okay, group in the middle. You want to try? Sliding filament theory of muscle contraction. So the sliding filament theory of muscle contraction. Okay, so let me orient you to what we're going to be talking about in number 1. This whole part right here is trying to get an action potential to start on the muscle, but don't you have to actually start on a neuron first before you get the muscle excited? Yes? Okay, so first thing that we're going to have to do is get the neuron to have an action potential, and then the in-between, and then get the muscle to have an action potential. Does that make sense? Okay, so you guys are next. Scott's group over there. Okay, I'm actually going to do B for you because I didn't talk to you guys about it much, so you're going to end up doing C. All right, so here we go. We are talking about an action potential, and it's going to arrive at the end of the axon on a somatic motor neuron. Everybody oriented to what we're talking about? Okay. And that is going to cause those new channels that you just met today called voltage-gated calcium channels to open on the synaptic terminals. Everybody good with that? Okay. And then C is what I would like for Scott's group to answer. After you open those voltage-gated calcium channels, it causes something to be released from the synaptic terminals. What is released from the synaptic terminals, guys? Nope. It's acetylcholine is released from the synaptic terminals into, you guys are next, over here, okay, this group right here, into the synapse between a neuron and a muscle called the blank junction. Neuromuscular junction, you got it. Into the neuromuscular junction. And now we are at D. Who's my next group? Anna, is that you? Okay, let me read it through so you'll understand what we're looking for. The thing that we just released will bind to something on a portion of the motor muscle cell membrane. Okay, first question is D, what did we just release? Acetylcholine, good. And Alice, acetylcholine is probably going to bind to what? What does things bind to? Nah. Acetylcholine is going to bind to what? Do you guys? It's going to bind to it. Say it.
I don't know what type of receptor.
It doesn't matter. I wasn't specific. Just, yes. Acetylcholine is going to bind to receptors on, this is you guys, the blank, blank, blank portion of the muscle cell membrane to give you, it's the curvy portion with all the receptors on it. What's it called? Motor end plate portion of the muscle cell membrane. Yes? Which is also called the sarcolemma. Everybody with me? Motor end plate. Yes. Okay. Everybody good? Okay, take a deep breath. We're going to go again. Back to the back. Okay. F is you guys again. Glacia, your group. F. wait, where are we on now? E, sorry. E. These protein receptors are also acetylcholine-gated what ion channels? Do you guys know? Acetylcholine-gated ion channels. What kind of ion? Karina, do you know what kind of ion channels they are? On the muscle cell membrane, the acetylcholine receptors are also ligand-gated blank channels. Karina, do you know?
Sorry, I was still asking on these and I got lost on it there.
Okay, it's right here.
Oh.
E. These protein receptors are also acetylcholine-gated what channels? Okay, you want to help them? Sodium. Yes, they are acetylcholine-gated sodium channels. All right. Okay. Don't worry about writing these down, guys. It's going to be on the video. So just freaking listen. Okay? It's gonna be on the video. So engage brain instead of pen. Got it? All right. So here's where we are. These protein receptors are also acetylcholine-gated what channels? Sodium channels. Okay. Now, do you guys feel confident about answering this one? F. Listen. Opening these channels causes a blank polarizing graded potential and excitation of the sarcolemma. If you open a sodium channel, you cause a blank polarizing graded potential. What kind of graded potential do you cause? Blank polarizing. It's a little bitty blank, hint, hint. Depolarizing. Say it with confidence. A depolarizing graded potential on the muscle cell membrane. Depolarizing graded potential. Okay. We are coming back to Scott's group. If threshold is reached, if I hit threshold, the action potential, watch me, begins on the sarcolemma and travels deep into the muscle fiber via little blank, big blank. What is it? T-tubules, exactly. Okay. All right, so H is next. We are going to your group right here. Ready? Okay. After it goes down the T-tubules, the action potential arrives at the place that stores the calcium. What is that place? Yeah, sarcoplasmic reticulum. Everybody good? Sarcoplasmic reticulum. Okay. Now, Anna, your group. Action potential causes what to be released from the SR? Calcium to be released from the SR. Good? Everybody good? Okay. Now, calcium, you guys, calcium binds to the sarcomere at a little bitty protein fiber. It's the switch for the barricade. What's that protein fiber called? Troponin. Yes? Troponin. Yes. Troponin. Okay. With calcium bound to troponin, the barricade protein. Raise your hand if your group knows the name of this protein. The barricade protein, okay, Karina. The barricade protein what? Tropomyosin, which prevents cross-bridge formation, is lowered. Okay, now that we're going to do this like 3,000 times. Which protein has the heads, people? Myosin. The myosin heads can now contact which protein has the active sites? Actin active sites. Every single time you hear heads, it's myosin. Every single time you hear active sites, it's actin. So keep that in mind because it's going to be repetitive throughout the rest of this. Active sites. When the heads and the active sites are bound to one another, what's it called when you do this? You form a cross bridge. Good. Now, you guys know this part. Flappy, flappy. To initiate a power stroke, what must happen? What must fall off? Phosphate has to fall off. What protein has the heads again? Everyone. Myosin head. The myosin head now pivots for the first power stroke. Good. At the end of the power stroke, what has to fall off? ADP falls off. Good. Now we are going to detach the cross bridge. Okay. One more time. What protein has the heads? Myosin heads must release what protein has the active sites? Actin active sites. And reset for another power stroke. This requires what? ATP, yes? Okay, got it? Okay. Okay, so G. Raise your hand if your group knows this one. For a sarcomere to shorten, this attach, power stroke, detach, reset, whole business must happen over and over again, right? This repetitive process is a cycle, and it's called, raise your hand if your group knows it. Go ahead. Cross-bridge cycling. Yes? Got it? Okay. Now we're going to get back to the new stuff, which is how do you make a fiber relax? Okay? How do you make a fiber relax? So, in order for the muscle fiber to relax, so listen to what I'm talking about so that you can see. So, in order for a muscle fiber to relax, we have to stop having an action potential on this thing so that we no longer release acetylcholine. So I'm backing up. Before I get to the muscle, I have to stop stimulating it. So in order for the muscle fiber to relax, the action potential on blank must end or acetylcholine would continue to be released. Raise your hand if your group knows that one. Go ahead. Go ahead. Axon or neuron, right? But you got to talk about the nervous system, right? It's the nervous system that releases acetylcholine. Got it? Okay. So the action potential on the axon must end or acetylcholine will continue to be released. Okay. Now we have to deal with the acetylcholine that was already present. You with me? So we are at B. The acetylcholine that was already in the neuromuscular junction must be destroyed or the muscle receptors would continue to detect it. The enzyme blank destroys acetylcholine at the neuromuscular junction. Please raise your hand if you know that one. Glaucia? No? Go ahead. Acetylcholine esterase, yeah? Or acetylcholine erase. That works too, right? Okay. So acetylcholine esterase destroys acetylcholine at the neuromuscular junction. Okay. Here we go. This one is stuff you've known forever. If you don't have a depolarizing graded potential, you can't get a blank blank. Raise your hand if you are confident and you know it. Go ahead. No depolarizing graded potential, no action potential. Yes? You've known that for ages? Okay. No action potential on the sarcolemma means that what will not be released from the SR? Again, it's the only thing that's stored in the SR. Somebody answer who has not talked. Calcium. There we go. Good. No, calcium will not be released from the SR. Okay. E should be easy. Listen to the whole thing before you try to answer it. Calcium present in the sarcomere must be removed for the what barricade to go back up? Raise your hand if you know it, especially if I haven't heard from you before. Speak to me if I have not heard from you before. I'm a friendly person. Go ahead. Thank you. Tropomyosin. Barricades to go back up. This is accomplished by pumping calcium back into the SR. Every single time you use a pump, you use what? No, don't answer it. I want somebody to raise their hand who has not spoken. Yes. ATP. Good. Requires ATP. Okay. Now we can relax. Now, which protein is the thin filament protein with the active sites? Actin, thin filament, slip back into place and the sarcomere lengthens. Whew. Deep breath. Cleansing breath. Everyone's okay. Okay, so this thing right here, guys, this is almost all of the complexities of muscle contraction and relaxation. And I made it for you, okay? But I want to tell you something about what you should probably be doing as far as studying. One of the things that I ask you guys to consider is to tell me how you study, what you use for study techniques. And I want to tell you what you said, and then I'm going to tell you what the literature says actually works. Okay, so what you guys said that you mostly did was reread your notes and your text and review your lecture recordings. Some of you did repetition memory. Some of you studied with a friend, rewrote your notes, created your own study materials. Guess which of these, any idea which of these is most correlated in actual studies with data to student success? Any idea? It is the stuff that you are not doing. It is creating your own study materials. So like that worksheet, you guys should be making them for yourselves, right? And the other thing that correlates is making up questions to test yourself. A students tend to do those two things, and the other things that A students tend to do is study with a friend or study group. This is not so much about the socialization angle, but getting you to use the terminology comfortably, right? So the things that don't work very well are flashcards, and this has no correlation at all to success. Highlighting things doesn't do anything, right? You just like end up highlighting the whole book. You're like, well, now I have a yellow book. Thanks. So we will talk about this more as we go through. I want to talk to you guys, but one of the things that it seems that occurs when you make your own study materials and then test yourself is that you take the knowledge from being mine to being yours, and then you critically evaluate what you know and don't know, which is the point at which you need to get if you're going to really do well, especially in phys, because phys is kind of complicated. Okay, so we are going to move to F, which is skeletal muscle energy metabolism. You can get away from your partners if you choose. It's fine. You can go back to your own space. Okay. So students very often ask me, why don't you have worksheets for everything? And there's two answers. I haven't written them. And, well, three. You hate them, like all that anxiety. And also, you need to make them for yourself in order for them to be super duper effective. Okay, so here's what we're going to talk about now. We're going to talk about how a skeletal muscle gets all that ATP that it needs both for cross-bridge cycling and also even for relaxation, right? So boatloads of ATP is needed. Everybody good with that? Huh? Yeah, only a little bit. You're like, I thought we were done with cellular respiration. You will literally never be done with cellular respiration until you're dead. But it's the same story, you know, with a couple of little extras because skeletal muscle does a couple of little extra things. So here's my question to you. A skeletal muscle skeletal muscle fiber and contraction keeps you from getting hit by a bus or eaten by a tiger or whatever the hell, right? So my question is, how does skeletal muscle make sure that it's always instantaneously responsive, right? So let's just say the skeletal muscle was sitting there and you hadn't eaten in 72 hours and the tiger's running at you. You can eat a Snickers bar, you're going to die, right? Because it's not going to get there fast enough. So not surprisingly, your skeletal muscle leaves several reserves behind so that you can always get quick access to a muscle contraction, right? So there's reserves in the skeletal muscle that it's preparing for a rainy day or a tiger. So let's look at the reserves and how they exist. Okay, so the first thing is that, remember, I always leave my myosin heads in the high energy position, yes? So that I can always get one cross-bridge formation and power stroke for free because I paid for it before, right? So I always get that for free, that first cross-bridge formation and power stroke, simply because remember the last thing that I do is hydrolyze the ATP to put it back into the high position, right, or back into the energized position. Does that make sense to you guys? So you always leave it there so that you always get that for free and that for free. It's not free, we just paid for it before, right? Okay, so, but that of course doesn't get you anything but one power stroke, so it's not particularly impressive. The next thing that you have inside skeletal muscle, but not all tissues, is you have this thing called creatine phosphate. You don't store a ton of it, but you do store some creatine phosphate inside skeletal muscle. And what it can do is take used ATP, which is ADP, and quickly, via one chemical reaction, and rephosphorylate it so you got another ATP. Does that make sense? And that's faster than cellular respiration, right? If cellular respiration was one chemical reaction, you wouldn't all hate it so much. Because then you'd be like, I got this, it's fine. So what you do with creatine phosphate is you rephosphorylate ADP to ATP. But you don't have very much creatine phosphate. You have enough creatine phosphate to do that for a minute of brisk walking, getting ATP, or 6 seconds of sprinting or lifting heavy stuff, right? So it's just a little creatine phosphate. By the way, the enzyme needed to do that is called creatine phosphokinase, or sometimes creatine kinase, abbreviated CK or CPK. Just a second, let me finish the story. And CK or CPK can be used diagnostically because although we're not talking about skeletal muscle or cardiac muscle right now, cardiac muscle has CK, CPK in it too. It also has troponin. So have you ever heard that when they think someone had a heart attack, they can do a cardiac enzyme test? Because when you damage the cell, the stuff that was inside leaks into the extracellular fluid and you can pick it up in your bloodstream. One of the things that they look for is cardiac CK, creatine kinase. It says, hey, you killed the cell. They also look for cardiac troponin, which falls off and leaks into the extracellular fluid and gets picked up by the bloodstream. Go ahead.
When weightlifters take creatine, is it actually helping them that much?
Of course, since it's a nutritional supplement, you know that there have not been clinical studies that have been done to a large degree with because, does it seem to work? Well, if you're doing like powerlifting, right? You don't need one more rep. I mean, you don't need 10 more reps. You need one more rep, right? Can this help provide that? It can, at least theoretically. Is it going to help at all if you're a cyclist? No, it's not going to help. But the other thing is that, two things about creatine kinase and supplementation, because they do sell this in health food stores and that stuff, and people take it for building muscle mass. The other thing, two things. It's water soluble and tends to make you retain water. So the weight gain, some of that is water gain, right? Okay, so it's a little bit of a confusion as to what's actually happening. It's kind of hard on your kidneys because the excess has to be cleared through your kidneys. So you have to drink lots of water and hopefully you don't have a kidney problem. And then third, this is not the only place that creatine phosphate goes in the body. And since we haven't actually assessed clinically or what the long-term effects of supplementing creatine kinase do, or creatine phosphate do, I want to tell you that it goes to the testes, it goes to cardiac muscle, it goes to skeletal muscle, and it goes to the retina of the eye. And I don't want to be the guinea pig that experiments with any of those things, right? So I wouldn't use it. And I would really discourage my 17-year-old from using it to try to build muscle mass because you don't want to be the person that figures out that, hey, in 25 years it causes testicular atrophy or whatever the hell. I don't know. So don't know. Okay, so creatine, so that's only used for like 1 minute, 6 seconds to 1 minute. So we're only like a minute in right now. Okay, and then please also remember that you do store a small quantity of oxygen so that you could have access to cellular respiration, aerobic cellular respiration, even before your respiratory and cardiovascular system caught up. Because if you think about it, don't you start working out before your heart rate and your respiration goes up? Right, they just have to catch up. So I can do some aerobic cellular respiration because I always store a little bit of oxygen on what? Where do I store oxygen? Myoglobin inside skeletal muscle fiber. So I have a little bit of oxygen stored, right? And then in addition, remember that skeletal muscle has its own little piggy bank of glycogen, right? So you've got glycogen stored inside skeletal muscle. I'm not talking about glycogen that's stored in the liver. That's different glycogen. This one has its own little piggy bank of glycogen. So I have a little bit of this stored and a little bit of that stored. And so typically, now this is really variable depending on what you've eaten and what you do with your body, but typically the glycogen that is stored inside the skeletal muscle fiber can get you 5 to 10 minutes of activity as your cardiovascular and respiratory system is catching up. So now we are 11 minutes or so in. Does that make sense? Go ahead.
Is the O2 bound in myoglobin an example of anticipatory homeostasis in a sense?
Well, if you're anticipating it constantly, then yeah. Because you're always anticipating needing oxygen, so you're using it. It's just stored there.
It can't increase based on if
Can it
you do a lot
increase?
of physical activity all the time?
Yes. I had never thought about that before. But once we get to muscle fibers, the types of muscle fibers, which is going to be the last thing that we do today, you can, with what you do with your skeletal muscle fiber, preferentially train it for aerobic activity, which results in more myoglobin and more capillary flow. So that was a good question. I'll get to it again when we talk about the different types. Fast twitch, slow twitch, oxidative glycolytic muscle fibers. Okay, so after we are, 10, 12 minutes in, hopefully my cardiovascular and respiratory system have caught up and now I am delivering blood glucose and delivering oxygen from the bloodstream, right? And then how long does this last me? Well, it depends on what I've eaten, of course, but let's say it can last you for maybe the next 30 minutes, your blood glucose, right? Okay, so now I'm 41 minutes in, right? Only then does the blood glucose usually run out and then you switch to burning fatty acids that are in your bloodstream, Okay, so then you switch to burning fatty acids for cellular respiration. Does that make sense to you guys? Now, am I saying that you will never ever burn a fatty acid or burn fat without working out for 41 minutes minimum? No. This cell has a whole bunch of reserves, so it doesn't switch over to fatty acids very quickly, but the rest of you can, right? And remember, fat storage is body-wide metabolism, not individual cell metabolism. So skeletal muscles have a lot of sources before they get to fatty acids, but it's not as if the rest of your body is not going to use that. Okay, so now let's just reinforce and then we'll talk about lactic acid. So you have known this for ages. I want to think about what requires oxygen and what doesn't. So take glycogen and break it down to glucose. That's glycogenolysis. Does that require oxygen, guys? No. Take glucose, break it down to pyruvate and ATP. That's glycolysis. Does that require oxygen? No. Glycolysis occurs in the cytoplasm and not in the mitochondria. It doesn't require oxygen. Okay. Now, plan A would be now I have oxygen and I can go through Krebs and oxphos, right, and get boatloads of ATP, right? But your skeletal muscle still has a plan B because if a tiger's coming at you and you don't have any oxygen, you're not going to go, it was a nice run. Hi, tiger. So you actually can do a little bit of anaerobic cellular respiration in skeletal muscle, but it doesn't generate very much ATP and it generates a byproduct which will cause fatigue, which is called lactic acid. So let's look at what happens there. Okay, so we already know these. Breakdown of glycogen doesn't require oxygen. Glycolysis doesn't require oxygen. Anaerobic respiration requires no oxygen and produces a small amount of ATP. Now, you have probably been told, like I have, that lactic acid causes muscle soreness, and I would be willing to bet that half the trainers still teach that, but it's actually not true. Lactic acid does tend to correlate with muscle soreness, yes, but correlation is not necessarily causation. So lactic acid changes the pH of the muscle, and that's part of the reason they think it ends up with muscle fatigue. They also think it might actually just get in the way of sarcomere contraction. But lactic acid is associated with muscle fatigue, especially since you're not making very much ATP, right? It tends to correlate with muscle soreness simply because if you use your muscles really massively and don't provide them with a lot of oxygen, you can get connective tissue strain, which is the reason for muscle soreness. Okay, they've actually done some experiments in which you really strain a muscle but suffuse it with oxygen, and then it gets soreness without lactic acid. And so you've uncoupled correlation and causation, right? So you say they occur at the same time, but they don't. It's not necessarily causative. Okay. So Krebs and oxphos, if you want to go through all of those are going to require oxygen, of course. Okay, so now the last thing about this is you guys have probably heard many times that when you work out your muscles, your metabolism is not just high while you're working out, it's high for hours afterward. Have you guys heard that? And it makes total sense if you remember that you need to put everything back that you just used, right? And most of those are either oxygen requiring or anabolic reactions that you are going to do. So you are going to continue to need ATP, oxygen, and potentially glucose after your muscles are used so that they're ready to use the next time. So let's see what you have to do afterward. The oxygen that was bound to myoglobin and also hemoglobin has to be replaced because I just used it, which means that my respiratory system, although theoretically it could go down immediately, does it? No. And your cardiovascular system could turn down immediately too, but does it? No, right? It takes a few minutes. Could you not just turn it down with the nervous system? You totally could, but you don't need to at this point. And the creatine phosphate, guys, you have to put it back. So you're finished using your muscle for contraction, and I need to make more creatine phosphate so that I can get quick access to rephosphorylating ATP next time. Where do you think I get the phosphate to rephosphorylate creatine phosphate? Creatine phosphate. Where was it? Where do you think I get the phosphate to rephosphorylate creatine to creatine phosphate? Who else? No, where do I steal that phosphate from? Now that I'm no longer contracting, I can steal a phosphate from ATP, guys. Yeah, I steal it from ATP, rephosphorylate. Why did I do that? Well, I'm not using it for contraction anymore, right? I'm going to leave it behind so that I can have quick rephosphorylation the next time I'm doing contraction. So that means that this is an energetically expensive rephosphorylation process. Okay, so the next thing is, after I'm done, I also need to get rid of that lactic acid. I take lactic acid and I oxidize it back to glucose. This happens in the liver and it requires oxygen. So again, cardiovascular respiratory system still shouldn't turn down yet. If there was lactic acid generated and dumped, I need to reoxidize it. What was your question, Mike?
So in order to, with creatine, in order to make more ATP, you have to use ATP.
Yes.
And then to store ATP, you need for creatine as well. So it's all.
Yeah, but so why is it worth it to do this, by the way? I mean, it's kind of a good question. Like, why is it worth it for you to spend ATP to make creatine phosphate? So what's the real advantage to having this here? Any idea? Because it's a quick access to ATP for that time in between when you haven't actually got enough oxygen in to generate your own ATP. So this is really just like this in-between space, and you're using it for like a minute or so as everything else is ramping up to serve other purposes. But I do need it, right? I need it in that in-between time in which I don't have access to other things. Go ahead, Alice.
So are you rephosphorylating creatine the entire time, or are you doing that after you're done exerting energy?
After.
So you wouldn't do that at all until you're like not moving anymore?
I have never heard that it is going on during muscle contraction. It is post-muscle contraction as far as I know. Post-muscle contraction. You wouldn't want to use ATP for that purpose during muscle contraction, right? It's after muscle contraction. So this is when you're sitting around thinking about what a boss you are because you worked out at 6 o'clock in the morning, right? And you give yourself a donut because you deserve one, right? And I'm going to make it into some creatine phosphate is what I'm going to do with it. Does that make sense? Okay. All right. So I took also, where was I? Lactic acid has to be oxidized back to glucose by the liver. That requires oxygen. Replacing all of these things are either oxygen requiring or anabolic reactions that require ATP. So you got a time after you work out in which your metabolism is still going to be really high. That seems to, in a lot of people, correlate to if you work out in the morning, you wiggle, your metabolism stays high, which means that you end up wiggling more over the course of the day than people who don't work out in the morning. If you work out at 6 o'clock in the morning, I admire the hell out of you, and we don't have a whole hell of a lot in common. But this is also the reason, have you guys ever tried to work out right before you go to bed? Your metabolism stays so high that you can't really sleep a lot of times. It is high. Your metabolism still is high. So you continue to breathe deeply and rapidly, and your cardiovascular system doesn't slow down as quickly as it seems like it would. You are repaying, they call it repaying the oxygen debt, but it's bigger than oxygen, right? It's glucose to make more glycogen, it's oxygen, it's ATP, right? You're just putting everything back. Why? Because there could be a tiger at any minute. You need to be prepared for tigers trying to eat you or buses trying to run you over, right? So you always, like at the end, put all your toys back, right? So it's a good little muscle cell. Put all the toys back. The other thing is, of course, it increases your metabolism afterward because your body just heated up and now you're going to have to expend energy to cool it back down, right? That makes sense? So your body heated up and it's energetically expensive to cool it down. So homeostasis, restoring your homeostatic body temperature. Go ahead.
I was just thinking, like, so this way, you just think about professional athletes and stuff like that, where hyperbaric chambers, this is how they, is this just a...
They are just suffusing with 100% oxygen into their tissues. Yeah. And it can help with recovery. It can do a lot of things. It's probably not as effective as they think it is, but maybe the placebo effect is like, now I'm amazing. The other thing, by the way, the reason, do you guys remember that energy conversions tend to be really inefficient? So what I'm going to do with muscle contraction is take chemical energy and ATP and convert it into mechanical energy for cross-bridge cycling. That tends to be inefficient. Any energy conversion is inefficient, so you tend to release a lot of heat as a byproduct. That is one of the reasons that muscle contraction and relaxation is one of the most dependable ways to heat up your body. That's the basis of shivering. I'm going to do this energy conversion, which is always really inefficient and always generates heat as a byproduct. So when you're cold, you shiver, right? Because it heats up your body really dependably. Everybody good? Yes? No? Do you want to talk about cellular respiration more? I know it's your favorite topic in all of physiology. It's Anna's favorite. Okay, so the next thing we're going to do is the stuff that you did in lab, which is talk about muscle twitches and talk about how you can get a muscle fiber to contract more. Okay, so this will correlate directly to the stuff that we just did if you let it. The response of a single muscle fiber to a single stimulus is called a twitch. Okay, this is one muscle fiber. It doesn't mean this. This is not twitching, right? It's one muscle fiber. You stimulate one muscle fiber and it has one response, and that is what a twitch is. Does that make sense to you guys? Okay, so there are 3 phases to the twitch, and you learn these in lab, right? Latent period, yes. Contraction phase, relaxation phase. That make sense? Okay, so the latent period, I want you guys to put together what is happening. This is the period of time when it was stimulated, either electrically or with acetylcholine released, but before it actually has responded. So here's my question to you. What is happening inside the muscle cell during the latent period? Go ahead.
Is it starting to generate tension?
It is, but how? What are all the in-between parts before you even start to generate tension? So I've got an electrical stimulus, but I have no contraction. What happens in between? Electrical stimulus. What does it have to do? Has to go down the T-tubules, and then what has to happen? Calcium has to be released, still haven't contracted yet, right? Calcium has to bind to troponin, and then tropomyosin has to move out of the way. Only then do I form the first cross bridge, right? So all of that we talked about is all happening right there in the latent period. Can you understand why the response isn't instantaneous? Because there's some crap that has to happen, and some of it is diffusion based, and you can't make diffusion go faster than it does unless you want to heat it up. Okay, and then here's the contraction phase, guys. What is happening inside the sarcomere during the contraction phase? Yep. The cross-bridge cycling. You're sliding the filaments, right? That's the sliding filament theory. And last question, I'll stop torturing you about things that I just talked about and you don't want to talk about anymore. The relaxation phase. What's happening to allow the muscle to relax? Tell me how you get it to relax inside the muscle fiber. Get rid of calcium, yes, and then what happens? Tropomyosin goes back into position, block the active sites. What slides back? What's the protein that slides back? Actin slides back. Got it? Okay, so although it didn't seem like it when you did it in lab, this is directly related to the physiology that you learned in lecture. Okay, so let's just do a couple of things and talk about the contractions that you learned in lab. So tension is what you generate with the muscle, and load is the thing that you're trying to overcome. So if I generate tension, I'm generating tension with my biceps brachii, my brachialis muscle, right? And the load that I am moving is simply basically the mass and gravity of my forearm, right? But of course I can increase the load, yeah? You can increase the load. Now, tension and load oppose one another. If the tension overcomes the load, then you'll have measurable shortening. If the tension doesn't overcome the load, do you still have cross-bridge cycling? Yeah, you do, right? Because like right now, I'm tensing my abs and I'm generating tension, but I'm not overcoming the load, right? Okay, so you still microscopically are going to have action happening. You might not have measurable shortening. Does that make sense? So that's tension and load. So let's look at the type of contractions that you guys did in lab. An isometric contraction seems kind of counterintuitive, but it's not really. So with an isometric contraction, you can see that what you're doing is with this, they have 2 rigid supports, so the muscle's not going to move, right? And I'm going to stimulate it. This time I'm going to do it artificially like I did with my frog, gastrocnemius. I'm going to stimulate it with an electrical signal, cause an action potential, calcium release, right? And it's going to generate tension, but it can't overcome the load. So this is an isometric contraction. Iso meaning same, metric implying length, right? Seems counterintuitive that you can have a contraction that stays the same length, but you are doing them right now, right? You are holding your neck up, right? You are holding your body in position. You do a lot of isometric contractions, just holding postural stability. If you ever do yoga, you like put yourself in a position and hold it, or Pilates, you do the same thing. So isometric contractions are contractions in which cross-bridge cycling is occurring, but the tension is not overcoming the load. Now sometimes it's a forced isometric contraction. Like if I try to do this, I can't overcome the load, right? So that's forced isometric contraction, but it's still isometric, right? Okay. Now, isotonic is the one that I had the hardest time understanding, like why it was called what it was called, but I want you to understand it. Okay. So an isotonic muscle contraction, I'm going to set it up similarly, only I'm going to make the bottom one a movable lever with a load that I know that I can overcome, right? So you have to experiment a little bit to see if it's a load that you can overcome. So I've got it set up, an electrode, load on the bottom. I'm going to stimulate it. And what's going to happen is it's going to start out isometric, right? But then if you can generate enough tension to overcome the load, it will end up shortening. So the reason this is called isotonic, this is what I finally put in my head, and maybe it'll make sense to you. Isotonic means same tone, right? So what's going to happen is you are going to generate tension, 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 but then as soon as you generate enough tension to overcome the load, you don't generate more tension, you actually just shorten, right? So this is actually movement. So what happens is once you get the tone enough, the tone stays relatively constant. You don't have to continue to increase the tone. So this one actually wiggles you, right? So generate enough tension and then you shorten. And then the tension goes back down. So isotonic, meaning that once you get the tone enough, you will actually do movement. So an isotonic contraction is like me doing this, right? And interestingly, so what's happening with that picture, if I lift something that I think I can lift, that's like almost outside of my capacity, what's going to happen right here is I'm increasing the tension, right? Increasing the tension. And then what happens here is that I have actually generated enough tension to overcome the load. Yes? But that's different for me than it is for some of you guys, right? So your capacity to generate tension differs with the amount of muscle mass that you have and the strength of your muscles, the training of your muscles. So this is a wiggling contraction, and it's called an isotonic contraction. Does that make sense to you guys? Go ahead.
Is that the same as tetanus contraction?
No, tetanic contractions are when you stimulate them really, really quickly. I'll teach those in just a second if I have time. If I don't, I'll do it on the video afterward. Okay, so now this one isn't in most textbooks, but it's kind of an interesting thing to think about. The concept of a lengthening contraction. And a lengthening contraction sounds counterintuitive. So with a lengthening contraction, what you do is, have you guys ever done a back bend as an adult? Like in yoga? You do not do it like you did when you were 8, because you will break something, and you know that, right? So what you do in yoga when you do a back bend is you completely engage your muscles, right? And then you stretch the origin and the insertion away from one another. And you are contracting, yes, but you are lengthening. But it's really just a subtype of which of the other two? Isotonic or isometric? Which one? Which one do you think it is?
You said you were shortening?
No, I'm with these muscles, right? So what I'm doing, I want to know what I'm doing with my abdominal muscles when I do a back bend, right? So I engage them, yeah, and then I stretch them. It's a subtype of an isometric contraction. Why do you engage them at all? Well, because you're going to break something if you don't. You're trying to protect the joint, right? Now in the back, right, what I'm really using is these muscles right here to primarily do the activity. These are doing isotonic contractions. These are doing engaged isometric contractions, which is called a lengthening contraction. And the reason you do those is because they tend to pull, give more flexibility to the connective tissue after you've done that. Okay, so the thing that Scott was talking about, I want to talk about briefly, and I will go over it again on the video, so if it's rushed here, don't worry. Okay, so I want to talk about how you get one muscle fiber to contract minimally, maximally, or somewhere in between. And this is called summation, and you guys did this in lab. And it really, really correlates to the idea that we learned about how do you communicate intensity with the nervous system? How do you communicate intensity with the nervous system? Frequency coding. Okay, so how do you communicate to a single muscle fiber that you want it to contract minimally, maximally, or somewhere in between? Frequency. So here's what happens, guys. If I stimulate a muscle fiber once, I get one twitch, yes? Okay. What if I stimulate it and then do not let it completely relax before I stimulate it again, right? Then what I'm getting is summation of calcium release, right? So what I let it do is release calcium, start a contraction, zap it again, release more calcium, so I get a stronger contraction. Zap it again, release more calcium, so I get a stronger contraction. That means that this muscle would hardly contract at all. This one would contract more. This is one muscle fiber. This one, if you stimulate it more frequently still, you can end up getting a relatively high average tension, and this is called incomplete or unfused tetanus. Does that make sense to you guys? So what is happening when I'm like holding up something? Pretend this was way heavier. If I'm holding up something that's relatively heavy, what's happening is my nervous system is sending relatively frequent stimuli to maintain a high degree of contraction, but it's not doing this, guys. It's not doing complete tetanus to the point that I am sending them so frequently that there's no relaxation at all. And this actually doesn't happen very frequently in a person who's not being electrocuted or have an ion imbalance. If you get into this maximal tetanus in which you're holding something like really, or get electrocuted, your muscle goes, and it's really, really painful because it really yanks on your connective tissue maximally, and you can't sustain it for very long either. It's going to completely burn up all the ATP, and so you won't be able to continue doing that. So that's complete or fused tetanus. So your nervous system will determine how frequently you need to send a stimulus in order to get that muscle fiber to contract more. And then in the video, the last part of this, I'm going to do like a 20 or 30 minute video to finish up muscle. I'll teach you how you control whole muscle contraction, right? So again, the plan is today or tomorrow, I'm going to finish up this with a video, right? And the video is going to go through the last little part of muscle. I will test more easily and kindly on the stuff that we didn't have the opportunity for questions about. It's going to cover whole muscle contraction and just a little bit about smooth muscle. And then you're going to have a quiz, and the quiz I hope is going to be posted tonight or tomorrow. And the quiz will be a review quiz over everything, including that video, right? It is open book. I encourage collaboration. It's supposed to assess whether you're ready for your test. Your test on Tuesday will be over endocrine system and muscle. Good? Okay. Quiz will be due. I'm going to make the quiz due. I'll tell you on the announcement, but I think I'm going to make it due Sunday night so you can talk to each other Monday if you got something wrong. Because I know a lot of you guys are going out of town, but you'll probably be back on Monday.
So my notes are a little different. I put them on Wednesday night.
I think I just changed them, and they're not, the order is just different, just a tiny bit different.
Okay.
I didn't mean to change the one that you guys were looking at, but I accidentally did, and I don't know what that looks
It
like.
doesn't have that one big option link for me.
Contractions?
Like, the contraction.
Okay. It's nothing major, I don't ever ask about that anyway.
Okay.
I realized that in the last class, I didn't realize that I changed the one that I was going to, I thought I was changing the one for next one. Yeah.
Are you going to
Oh, this I have to do for that,
class?
right? They have their lab and stuff.
Like that.
So you've already had your midterm now, Thursdays.
Okay.
And then, so when
I don't even like being in California. It's difficult to I never have. Yeah. I like every single family photo. I'm not trying to be a good person, but like, you guys all enjoy this and I hate it. It causes me such anxiety and has to know it. And it doesn't matter, like, in retrospect, oh my God! Latex, that in the fee. Motor unit recruitment. So this has to do with how much you stimulate a single one. Motor unit recruitment has to do with down his flights.
And that's like, so if I pick up a laptop and I can't move, like, you know,
So, you can get certain personnel, you will be going under the but then what's going to happen if I try to pick up your backpack? So what's going to happen is I'm going to look at it and say, hey, my subconscious goes, I know what background changes way, and it will recruit a minimum number that might do the job. And then as I realize that you got freaking rocks in here, it continues to recruit more units until you get to the point that the tension you generate becomes low. So you're doing both of them at the same time, both going more frequently and then more cells. Right? No, summation is always holding it to a single cell. Oh, when I'm saying one fiber. You can control what one fiber does and then you only maximally or somewhere in between. But then the other way you can control it, you can pick something up, is how many fibers do you use.
Like the picture of like a whole muscle, like little tiny one, opens up bigger and bigger.
Yes. And you don't have to use all of the fibers in your muscle. And if I try to pick up a pen or an iPad, my body goes, I know what something about this size probably weighs. And it says, let's just do a random number of motor units and see if that works. And if that works, that's totally great. But if I need to hold it up for a longer period of time, then I can continue to stimulate them more frequently, right? So I can have a high level of maximum tension, but I don't need any more cells. Does that make sense?
Yeah.
Right? So, yeah, so they're both going on, really. So one of them is a one-cell thing, and that's which summation, temporal summation, yeah. And then motor use recruitment is multiple cells. Yeah, and I'm going to go through that in the video.
I have a question about complete tetanus.
Uh-huh.
So why you said when that like stimulates something you're like completely contracted because of complete tetanus, that you can't really maintain that for very long because you don't have ATP.
It actually changes the pH of the muscle cell and makes it really hard for the sarcomere to hold on.
Okay.
That's why they think that it causes it to
I think that you would
massively.
get stuck like that because you don't have
You
the
do at first, but then it changes the pH. What they think is that if you change the pH, oxidation formation can't occur very effectively.
Okay.
Something changes about the protein conformation, all that lactic acid, like it's dumped in there, and then you're like, oh, I just can't even hold on to this. But that's
There's no relaxation either.
no relaxation at all. Right. And it burns it up and it generates a whole bunch of pH changes that makes it really hard to hold on to it too. So you like spaz out and hold it really tightly and then it has like let's go. And they think, and the last time I read, I don't even know if the answer to this is absolutely known, but I'm going to tell you what I think I remember reading. They think it is because the lactic acid buildup causes conformation changes in the muscle and then you just can't hold on. It's not permanent conformation changes in the muscle because pH changes just temporary. And then you'll dump the lactic acid and make it into something else or whatever. Well, I think I'll have to do this once. I bet I'll have to do it again next semester.
I'm trying to add but I will.
I hope it works. I'm not doing it again.
Well, send it to us. We'll watch it.
No!
What? Come on.
Hell no. Like, when they told me they were going to do this, I was like, it was 4 weeks into the project. And I was like, I don't know if I want to be involved in this project if there was videotaping going on. Like, I was like, I want to kind of drop out.
Are you gonna be here Thursday?
Thursday?
Yeah.
No, we have break.
I know
Fall
that.
break. No.
It's funny you mentioned those little stats. I'm sitting here stacking up
Now, it's not saying it's negative. It's just saying that, like, as far as the data looks like, it doesn't have any correlations to success. So I'm going to share more of this data with you as the semester goes
I
on.
know where you are, I can see you.
Because that's part of what I'm reading and studying with this project that I'm doing. And it was actually really surprising to me. Guess what correlates negatively to performance?
Memorizing?
No. Memorizing is fine depending on the class. It's more useful for anatomy than it is for physiology. But what has a negative correlation to performance in the class is doing the vocabulary and questions out of the textbook. It gives students some kind of sense that they know things, but the person who's writing their assessment is not the person who wrote those questions.
Right.
So it's just kind of giving them this false sense of confidence. and it correlates to poor performance, which is amazing considering the textbook authors put in so much money and time into it.
Yeah.
So I was like, what? It correlates negatively?
Yeah, I've kind of done like, so with